Bismillahirrahmanirrahim. Today we'll discuss major histocompatibility complex MHC class 2, and this is the second lecture of series of MHC. So today lecture is related with class 2, and we will discuss in this lecture structure and function of MHC class 2. MHC class 2 molecule are a class of major histocompatibility complex molecule normally found only on professional antigen-presenting cell, such as dendritic cell, phagocytic cell, some endothelial cell, thymic cell, epithelial cell, and B cells. By professional antigen-presenting cell, we mean that these cells are totally devoted to the immune system and take active part in the immunity. This is the dendritic cell, B cell, and this is the macrophages. They are professional immunity cells. These cells are important in initiating immune response. When they are infected or when they engulf or phagocytose any antigen, they stimulate the immune system. So they are important for the initiation of immune system. The antigen presented by class 2 peptide are derived from extracellular protein, not cytosolic as in MHC class 1. In this case, these cells phagocytose the external protein or the antigen or the bacteria viruses by phagocytosis and internalize these cell, and after that if they destroy the cell totally, so they remove it by exocytosis from the cell, or if they are not capable to destroy these cells, then they present these cells on the surface to the T lymphocyte for activation of the immune system, and by this way the immune system is activated and then clear this infected cell from the normal tissues. Loading of MHC2 molecule occur by phagocytosis. Extracellular protein are endocytosed, digested in the lysosome, and the resulting epitope peptide fragments are loaded onto MHC class 2 molecule prior to their migration to the cell surface. This is the mechanism. It proposes the cell phagocytose the bacteria and then internalize it. After internalization, add its enzyme which are present in the vesicle. They are proteolytic enzyme. They digest and break down the bacteria into small peptide chain, and these chains are then loaded to the MHC2 molecule, and after that they are presented on the surface of the cell for T lymphocyte to identify them and clear them. HLA in human, the MHC class 2 protein complex is encoded by the human leukocyte antigen gene complex HLA. Structure of MHC class 2 molecule. Like MHC class 1 molecule, class 2 molecule are also heterodyne, but in this case consist of two homogeneous peptide, an alpha and beta chain. Both the chains are heterodyne. They have alpha chain and beta chain, but in class 1 3 alpha chain, alpha 1, alpha 2 and alpha 3 are responsible, and one beta chain, but in class 2 there are two beta chain and two alpha chain. So the beta chain is made by 2 alpha and 2 beta helix. The alpha and beta region of the chain come together to make a membrane distal peptide binding domain. Alpha 1 and beta 1 make the distal portion of the chain, and alpha 2 and beta 2 make the proximal portion of the beta chain. Distal portion is away from the membrane and the proximal portion is near to the membrane. The antigen binding groove, where the antigen or peptide bind is made up of alpha helix and beta helix. This is the antigen binding portion. The distal portion is the antigen binding portion, and here is peptide binding clip, and this is made by alpha and beta domain, and as if you remember that in class 1, this portion is made by the alpha 1 and alpha 2, and in MHC1 there is an alpha 3 is invariable portion of the alpha 1 chain. Here the beta 2 is the invariable portion of the class 2 molecule of the major histocompatibility complex. Here we can see that in MHC1 the alpha chain was having the membrane segment. This is the transmembrane segment. Here you can see that the alpha chain in class 1 has the transmembrane segment, but the beta chain, you can see the alpha chain have the transmembrane segment and also the beta chain have a transmembrane segment. The antigen binding group of MHC2 molecule is open at both ends. Here you can see that the MHC molecule, this is the peptide binding clip, and it is open on both sides, on distal side and on proximal. Class 1 molecule is closed. Here you can see that it is closed on the distal portion. The antigen presenting by MHC class 1 molecule are longer, generally between 15 and 24 amino acid residues long. We know that the MHC1 molecule bind only 8 to 10 peptide long chain, but here the beta 2 can attach 15 to 24 amino acid residues long chain. Processing and presenting of extracellular or exogenous antigen. How the MHC2 complex present the antigen to the cell surface. This is an exogenous. In case of MHC1, endogenous antigens are presented to the cell surface, while in MHC2, exogenous antigens are presented to the surface of the cell. Extracellular or exogenous antigen originate from outside of a host cell. They are not directly infect the cell, but they are present outside of this cell, and like this bacteria which is lying outside of this cell, and this is called extracellular bacteria, and this extracellular bacteria is captured by its receptor, and then pseudopodiums are produced from the membrane of this cell, and it surrounds this bacteria and then internalize or phagocytose this bacteria into the inside of the cell. This is called phagosome. Here in the inside of the cell, secretory granule which contain enzymes which are digestive in nature, and these vesicles are called endosome. When here this is phagosome, and here is the vesicle which contain lysosome, and this vesicle is called endosome. Endosome contain enzyme which is called lysosome. So in the next step, lysosome attach with the phagosome, and when endosome is merged with the phagosome, release its lysosome enzyme to this phagosome, and this is now become endolysosome. This phagosome is become endolysosome. By adding its enzyme to this endolysosome, the enzyme lysosome causes breakdown of the bacteria into peptide fragments. When this activity is in progress, at the same time in the endoplasmic reticulum, there is synthesis of MHC2 molecule. The MHC2 molecule is synthesized in the nascent MHC class 2 protein in the endoplasmic reticulum when they are synthesized. So a chain which is called invariant chain, which consists of peptide, is added to the antigen binding site of the MHC2 molecule. This causes inhibition of attachment of this antigen binding site to the protein of the endoplasmic reticulum, and other protein which are present, either protein for the class 1 MHC. So these proteins are prevented from binding to the binding site of this MHC2 molecule. This is one function, and it also helps the MHC2 molecule in transportation from the rep endoplasmic reticulum to the Golgi apparatus. In Golgi apparatus, this invariant long chain is broken down by a protein which is called cathepsin. Invariant chain is broken down by cathepsin, and only a short or small portion of the invariant chain is still attached to the binding site of the MHC2 molecule, and this small portion is called CLIP, which is stand for class 2 associated invariant chain polypeptide. In the next step, MHC2 molecule attached with the CLIP is attached, transported to the endosome, and when this merge with the endosome, a CLIP portion is removed by a substance which is similar in structure to MHC2, and it is called HLADM. This removes the clip from the binding site of the MHC2 molecule. When this clip is removed from the MHC molecule, then it has higher affinity for the peptide which are broken by the lysosome enzyme in this phagosome, and these peptides are attached to the binding site of the MHC2 molecule. After this binding, the molecule is shifted to the surface of the cell, and then now this cell is become a target cell for the T lymphocyte. The T lymphocyte then attack this cell and kill it and remove it from the normal tissue. Now we will study it in magnifying. Here is the phagosome. It is called endosome. The bacteria in the cell membrane, it is called endosome or phagosome, which is phagocytosed by the cell. Here we have the lysosome, which contain enzyme, lysosome enzyme. This lysosome add with the endosome and make endolysosome. This fusion causes synthesis of endolysosome, and when this lysosome and phagosome merge with each other, the enzymes are released, and these enzymes break down the bacteria into small peptides. When these small peptides are generating also in the rough endoplasmic reticulum, synthesis of MHC2 molecule take place. This MHC2 molecule is to the binding site of MHC2 molecule along protein chain is added, and this prevents this MHC molecule to bind the protein of the endoplasmic reticulum and also protein present in the cytoplasm. The other function is this chain helps the MHC2 molecule in transportation from the endoplasmic reticulum to the Golgi apparatus. Here it is transported to the Golgi apparatus. In Golgi apparatus, this invariant chain is broken down into peptide, and when this chain leaves the Golgi apparatus, only a small portion of the chain is remain on the surface of the MHC2 molecule. This is called CLIP, which stands for class 2 associated invariant chain polypeptide. This again prevents the binding of cytoplasmic protein to the binding site of the MHC2 molecule. Then this CLIP and MHC2 molecule merge with the phagoendosome. So when this is added, the cliff is removed from the binding site of the MHC2 molecule, and the peptide are then attached to the binding site of the MHC2 molecule. After this attachment, the molecule is transported to the surface of the cell and presented to the lymphocyte cell. These lymphocytes then attack the peptide which are attached to the MHC2 molecules by a receptor which is present on the T cell. There is another receptor which is present on the T cell which is called CD4 or co-receptor, and this attached to the invariant chain of beta portion of the alpha portion of the MHC II molecule. This is called T cell receptor. This is for the peptide, and this co-receptor is for the alpha chain. So there is a tight binding between the cell and the T cell, and then by injecting or by these T helper cell then destroy the infected cell and remove it from the normal cell and clear it from the body. Here is a vision of the MHC molecule, MHC1 molecule. Here you can see the proteins are entered into the cell. These proteins are then broken down by the proteasome into peptide. These peptides are transported by TPA into the rough endoplasmic reticulum, and then they are attached to the MHC1 molecule. From MHC1 molecule, they are transported to the Golgi apparatus. From Golgi apparatus, they are added to the secretory vesicle, and the secretory vesicle exocytoses take these protein to the surface of the cell for the identification and attack by the T lymphocyte. So the difference between exogenous and endogenous transportation and processing is that these proteins, the viral protein, directly infect the cell, and these lung protein are broken down by the proteasome. And in the case of exogenous, the cell phagocytose internalize the bacteria, and the bacterial protein are then broken down by the enzyme which is present in the endosome of the vesicle, which are added to the phagosome, and these are broken down into the peptide chain. Other differentiation is that till the arrival of MCH2 molecule to the phagosome, it is inhibited by the presence of invariant chain. It is inhibited to capture other proteins. Here the chain from the Golgi apparatus, MHC1 molecule is directly transported to the cell membrane. In MHC1, the T lymphocyte the attack is by the CD8 lymphocyte or cytotoxic T lymphocyte. So thank you. Please subscribe the channel and press the bell icon for in-time notification, and leave your valuable comment for improvement of the channel.